I welcome you to this new lecture video. So today we are going to look at the physiology of the nervous system. So what I want you to understand here is that the physiology of the nervous system is going to give us an insight of how the body works. So there are conditions. Let's say, have you tried to wonder how you move your hands? Let's say how fast you move your hands in case you are in a condition where you want to react to something. Let's say something has bitten you on your skin, or you're touching an hot object, then you tend to withdraw your hand very fast. So the mechanism that is behind you moving your hand very fast and behind your muscle contracting very fast, that is now the mechanism that we are going to understand as we go through the nervous system. So it is going to give us an insight of what really happens at that cellular level that causes your hand or your muscle tissue to contract and to move the way it moves. So basically the nervous system is made up of two types of cells, just like the way we learned in anatomy. So the nervous system is made up of two types of cells. We have excitable cells and non-excitable cells. So in short, what I'm saying is that the nervous system is made up of the neurons and the neuroglial cells. And we said that these neurons, which are also known as nerve cells, are the excitable cells, which means that these neurons are specialized in a condition where they respond to a stimulus and they also generate an electrical signal that is known as an action potential or a graded potential. So these excitable cells are just specialized cells that can respond to a stimulus by generating an electrical potential or electrical signal that is known as an action potential, a graded potential. Then the non-excitable cells, which are known as the neuroglial cells, these cells we understood to say that these cells, they just help in supporting the neurons, because we had those cells that helps in the formation of the myelin sheath, that helps in the metabolism, and also that helps in other functions just to support the neuron, which are the nerve cells. So we are basically going to focus on the nerve cells because these are the excitable cells. Now how does these excitable cells help us in the movement of, let's say, your limb or in the contraction of a muscle? So we are going to understand now the base of that physiology. So first of all, before we dig into the physiology of these nerve cells, we also have to keep in mind that we already have the information or the idea about the body fluid compartments, and we know the concentration of those ions in each compartment, the extracellular, the intracellular. For example, we talked about, let's say on the extracellular as well as the intra. We talked about, we said that sodium is more on the extracellular compared to the intracellular. So we are going to find that sodium is going to be more on the extracellular and less into the intracellular. And we said that potassium is less in the extracellular and more on the intracellular. And we gave you the values that you are looking at. So all that knowledge is going to be applied here. So as we try to dig into the mechanism, let's try to understand a few terminologies that are going to guide us, that are going to help us create that roadmap that will make us understand the mechanisms. So as we all know, a membrane has got what we call channels, and these channels, they are known as ion channels. So these ion channels, they are the one that regulates the movement of these ions. So these ion channels, they are basically just specialized proteins that permit the influx or the efflux of ions. For example, if you are having a cell, let's say this is a cell that you're having. So on the cell membrane of this cell, it is going to have a channel, and this channel can be called let's say it can be a sodium channel or potassium leaking and so on and so forth. Potassium leaky channel and so on and so forth. So we have got this channel. So this channel will now regulate, or it will start permitting the influx or the efflux of these ions that are found in these particular fluid compartments. So we're going to find that it can allow the movement of sodium into the cell, which we are referring to as influx, or it can allow the movement of potassium out of the cell, which we are saying efflux. So when you say influx, it means that an ion will be moving inside. Then when you say efflux, it means that an ion will be moving outside. So ion channels, these are just specialized proteins that will be permitting the influx and the efflux of ions. Then apart from these ion channels, we also have what we call voltage dependent channel. So this membrane of the cell, it has what we call a voltage difference, which is referred to as the rest membrane potential, which is just the voltage difference across the cell membrane due to the distribution of these ions. So you're going to find that they're going to have a certain type of ion, like sodium will be more on the outside, potassium more on the inside. So due to the inequivalent distribution of these ions, they create a voltage difference that is known as the rest membrane potential. We are going to talk about it. So there are channels that we call voltage dependent channels. So these channels, they're also found on the cell membrane. So how these channels operate is that when there is a change in the rest membrane potential of this particular cell, these channels are going to respond by opening and allowing the influx or efflux of these ions. So they can allow either the entry of sodium, or they'll allow the efflux of potassium or sodium. Yeah, so these voltage dependent channel are that channel that opens depending on the voltage change across the cell membrane. Then the other term that you also have to know is the term that is known as depolarization. It is the term that is known as depolarization, spelled like this, depolarization. So basically, depolarization is just the decrease in the rest membrane potential of a cell from its normal rest membrane potential. So when we say decrease, let's say for example we're having a graph, and this graph, let's say here it is showing us, oh, this is the normal rest membrane potential of a cell, meaning that when a cell is at rest, its rest membrane potential, it gets to be 70 millivolts. So now when they say that a cell has depolarized, and we take this to be our 0, and then we take this to be maybe our positive 40, when we say that there is depolarization in that cell membrane of that particular cell, what we mean is that the resting membrane potential of that particular cell will start moving towards 0. So meaning that it will start moving towards 0 like this. So the movement of this resting membrane potential towards 0, that is what we call depolarization. That's why we are saying decrease. It is moving from where it is, like from negative 70 millivolts going towards 0. That is depolarization. Then repolarization is basically just the opposite of depolarization. So if it was moving towards 0, meaning that repolarization it means that the cell membrane potential will start going back to its normal resting state. So the movement of this membrane potential, or the decrease in the resting membrane potential, or the restoration in the resting membrane potential towards its normal resting state, that is what we call repolarization. So repolarization is just the restoration of the normal membrane potential of a cell. It is being restored to go back to its normal state or its normal condition. That is what we call repolarization. Then when we say hyperpolarization, what we mean is that as this resting membrane potential of this particular cell has been taken back to its resting state, when they say that now it has hyperpolarized, it means that it has now gone beyond its normal resting level. So if the normal resting membrane potential was negative 70 millivolts, when they say it has hyperpolarized, it means that it has gone beyond. So meaning it is going to become even more negative. Let's say negative 90 millivolts. Then it will reach somewhere there. So what we mean is that the temporal increase in the resting membrane potential, or the resting membrane potential becoming more negative, that is what we refer to hyperpolarization. It is becoming more negative than its normal resting state, or it is increasing from 70 going to 90, like that. Then when you say an action potential basically is just an electrical impulse that creates a basis for the conduction of signals. So this action potential, it can cause the membrane potential to move. So what I mean by that is that the action potential, how it is formed or how it comes about is when this resting membrane potential of a cell depolarizes, it will go and shoot just like the way we are seeing the graph going. Then it will come back to normal by repolarizing. Then again, after some time, due to stimuli, it will shoot. So the movement of that signal like that, or the conduction of signal like that, that is what we call an action potential. An electric impulse that is going to be conducted in that manner, that is an action potential. So it is an electrical impulse that will create the basis for the conduction of signal. So the signal going to be conducted in that particular manner. So the conduction of that, it is due to the same action potential. We are going to understand it further as we'll get some illustrations. Then we also have what we call the threshold potential. So the threshold potential, it is a level at which the membrane potential must reach in order to initiate an action potential. So let's say if this is the cell that we're having, okay, let me just use the same diagram. If this is the graph that is showing the resting membrane potential of a cell. So it means that for this resting membrane potential of a cell to fire an action potential or to initiate an action potential, it has to reach at a certain level. So at that point where it has to reach, that is what we call the threshold potential. Let's say if it is at negative 50 millivolts, it means that for it to fire an action potential, it has to depolarize up to negative 50 millivolts. When it reaches that negative 50 millivolts, then it will depolarize. So this is going to be a point that will mark now the initiation of an action potential. So a level at which the membrane potential must reach in order to trigger an action potential, that is what we call the threshold potential. Yeah, it's more like you are having a line. They tell you to say that what you are holding in your hands, you are not going to let go of it until you reach this line. So it means that when you carry, let's say you are carrying stones, when you reach that at that point, that's when you have to let go of them, then run. That point where you have to reach and drop your stones and run, that point is what we call the threshold potential. It is a point that is marking the initiation of you running. So if you are in a race, then you're competing with your friends, then they tell you to say you're going to carry your rocks in this pack or in this bag. So when you carry them, when you reach at that point, at that line which you have drawn, that's when you have to drop them and run now without the stones. So it means that at that line where you are going to reach will be a threshold potential. It will be a point that will mark you so that you would drop your stones and then continue your race without them. So basically that's the idea I want you to get for the threshold potential. A level at which the membrane potential must reach so that it can trigger an action potential. A line that you must reach so that you drop your rock so that you can now run freely very fast. Then when you talk about the other thing that you have to know is a principle. This principle is known as all or none principle. It is known as all or none principle. Sometimes you see it being written as all or none law. So this principle, it actually just tells us, or it states that an action potential when it is triggered, so meaning that when you reach your threshold, then that threshold point, there will be triggering of an action potential. That action potential will propagate without decrement. It will propagate without decrement. Decrement basically means that when an action, it means that without reducing its strength or without reducing in its wavelength or frequency, or without just it reducing. So this means that when an action potential is triggered, when a resting membrane potential depolarizes and reaches this threshold potential, then it triggers an action potential. That action potential that is triggered, it will propagate without decreasing or reducing in its amplitude or strength. It just has to be fired. So it means that whenever it reaches at that point, no matter what, the action potential must be initiated. That is what this law talks about, or principle talks about. All or none principle, telling you that when an action potential is triggered, it has to propagate without decreasing or without reducing in its amplitude or strength. So it's more like it is telling you that whenever the resting membrane potential depolarizes and reaches the threshold potential, the idea of triggering an action potential is non-negotiable. For as long as you have reached the threshold, the action potential must be triggered, must be initiated, it must be fired without even negotiating, not even at what, no. It just has to be triggered. So that is the idea of this principle. When an action potential is triggered, it has to propagate, or it propagates without decreasing or reducing its amplitude or strength. It just has to go. So, having talked about this, we also have to understand that there are certain types of channels that I would like you to understand or to know about before we get now into the main idea. So these channels are known as ligand-gated channels. So these channels are known as the ligand-gated channels. So this is how the ligand-gated channels, we can draw them. So assume that this is the membrane of a cell, and this membrane of the cell, it is having these channels that are known as the ligand channel. So these ligand-gated channels, how they work is that they are just going to open when a specific molecule binds onto their receptor. So this channel that you are seeing, this one here, that is the receptor. So this receptor, if you have seen it, is having a certain shape that is going to accommodate a certain type of a molecule. So let's say now you are having this molecule here, and this molecule goes and binds at this point, at this receptor of this channel. What will happen is that this molecule that is coming to bind there, it is also known as a ligand. So when this molecule binds there, this channel is going to open like the way we have seen here. The channel will open, and this channel will allow either the entry or the influx of certain ions. It can be sodium, it can be potassium, it can be chloride, it can be any ion. So when it opens, it will allow the influx, or it can also allow the efflux. That's how it works. So a ligand-gated channel, these are just channels that open when a specific molecule, which is known as a ligand, binds onto its receptor. That is how this ligand-gated channel works. Then you also have what we call leaky channels. So let's assume that this is a cell membrane that we're having. A cell membrane has got channels that are known as leaky channels. Let's say these are the leaky channels that we're having for the cell membrane that we have here. So these leaky channels, they are always open. They allow the movement of these ions that are inside. These ions that are inside, they can be negative or positively charged. They allow the movement of these ions down to their concentration gradient. So if you are going to find that we are having more cations inside of this cell and less of them here, these leaky channels, they will be supporting the movement of these cations down their concentration gradient, meaning they will be going to where they are in less concentration. So that's how these that's how these leaky channels, they work. They just allow the movement of these ions continuously down their concentration gradient. So if the ions are more concentrated on the outside, these leaky channels, they'll be allowing the influx of these ions that are more concentrated on the outside. So the driving force for these ions, it is just the concentration gradient, and now they are being helped by the leaky channel, which are always open. Then we also have what we call mechanically gated channels. Mechanically gated channels. So mechanically gated channels, these are just channels that open in response to physical deformation in the membrane due to pressure or stretch or vibration. So what happens is that if this is a cell, and this cell, there is a stretch in it, so that stretching, it is going to cause what we call these mechanically gated channel. This mechanically gated channel, due to the stretching of this cell, it is going to open. When it opens, it will allow the influx or the efflux of ion. And sometimes it can just be due to pressure. Maybe you are applying too much pressure on a body tissue, let's say on your muscle. You are going to find that your muscle will contract. But what is causing that contraction? Because that muscle has got a tissue, which is the muscle tissue itself has got cells. Now those cells, they are having channels that are opening, allowing the influx of these ion, and as well as the efflux of these ion, causing the tissue to respond in that manner. And sometimes it can just be due to vibration. So you're going to find that these mechanically gated channels, they open in response to a physical deformation of the membrane, meaning that when that pressure is applied here, it is going to cause a little bit of deformation in the membrane, or in the formation of the membrane, causing it to stretch and causing it to open these channels. This is how these mechanical gated channels work. So, having known or having talked about these concepts, when we now dig into what a membrane potential that we have been talking about, no, resting membrane potential is, we are going to now understand, because these are the terms that we are going to be using now. So I want you to pay attention so that you may understand, and also you may ask questions so that you can even understand them more. Okay, so let's now look at the concept of what a membrane potential is. What a membrane potential is. So, membrane potential is basically just like what I said, it is an electrical potential difference across the cell membrane. So that electrical potential difference across the cell membrane, that is what we call a membrane potential. So this membrane potential, it is just due to the unequal distribution of the cells, like the way we said. We know that in our body fluid compartments, we learned that potassium is more on the inside compared onto the outside, sodium is more on the outside compared onto the inside. So because there is that unequal distribution of these ions, they create what we call a potential difference or a voltage difference. That is now what brings about the membrane potential. So this membrane potential, being an electrical potential difference across the cell membrane, it has got factors that helps to maintain it, because we know that in as much as there is an equal distribution, these channels, we have channels like the leaky channels here, that just allows the movement of these ions, because we say that these leaky channels, they are ever open. So there are factors that helps to maintain the membrane potential, so that the difference, the voltage difference, can just be maintained at that particular level. So we have got specifically, since we are talking about a nerve cell, which is a neuron has got a resting membrane potential of about negative 70 millivolts. Yeah, about negative 70 millivolts. So now we are going to understand how is this negative 70 millivolts, which is a membrane potential of a neuron cell, maintained. What are the factors that are involved in the maintenance of these, the membrane potential of this nerve cell? So I'm going to use this diagram to show you how the membrane potential is maintained, how the resting membrane potential of a particular cell is maintained. So it is known as a resting membrane potential because at that point, it is the membrane potential that the cell gets to have when it is at its resting state. It is not excited, it is not hyperpolarized. By hyperpolarized, you just have to think like a cell is depressed to a point whereby for it to just feel good again, to feel happy, you just need something. You get it? So the resting membrane potential is how this cell gets to be when it is just at rest. It is not too happy and it is not too sad, but it is just there. It is just at its resting state. So that's the concept I want you to get here, and I want you to be working with. So let's now talk about how the whole thing happens. So what we are going to start with is what we call the first factor, which is, so the first factor that we are going to talk about, it is what we call the sodium potassium ATPase pump. These sodium potassium ATPase pump are basically just channels. These channels, they allow the pumping of ions into the inside of the cell and also outside of the cell. So they do so by the use of an energy molecule that is known as ATP. So how these sodium potassium ATPase pump work is that I want you to look at these channels. So these are the sodium ATPase pump. So as you have seen from the name, we are having sodium as well as potassium. So meaning that they are just involved in the pumping of sodium and also potassium. So what they do is that they are involved in the pumping of potassium. It will be pumped into inside of the cell, and sodium will be pumped into outside of the cell. So they'll be getting those sodium that are inside, will be taking them back. You are not supposed to be here, go back. Then the potassium that is on the outside, they'll be bringing them in, come back home. You're not supposed to be outside. So you'll find that they'll be pumping 3 sodium outside and 2 sodium inside. Now if you look, it is more like it is more like they tell you that you are going to be getting 2 coins from you, or rather we are going to be getting 3 coins from you, then we give you 2. Who's going to be making profit there? The one who's getting 3 and they are giving you 2. That shows that they are getting more and you are losing 1. So what will be happening here is that inside of the cell here, they'll be removing 2, they'll be removing 3 sodium outside and pumping in 2 potassium. So what happens here, from the knowledge from the fluid compartments, we know that sodium is more on the outside here. So you are going to find that already the concentration of sodium is high on the outside, but this pump still keeps on pumping more sodium outside. And we know that potassium on the inside is also high, but this cell keeps on pumping more potassium on the inside. So it is going to create what we call a chemical gradient, because it will be increasing the concentration of sodium on the outside as well as the sodium or potassium on the inside. And what will start happening is that the concentration of this potassium on the inside, according to its concentration gradient, you are going to find that it has to move onto the outside, because that's why it is in less concentration, and for sodium it will need to go on the inside, because that's why we're having less concentration of it. So that is the concentration gradient that is being formed here. So from the work of these channels, you're going to find that this channel will be pumping these ions against their concentration gradient, just like they have said, and it will be causing to have more potassium on the inside and to have more sodium on the inside. But more of these ions that are positively and charged are going to be taken out. As you have seen, more of sodium are being taken out and less of potassium is being taken in. So this is going to contribute to the rest membrane potential about -5 millivolts. About -5 millivolts. So the electronegativity of these rest membrane potential, since we said that the rest membrane potential of a nerve cell is about -70 millivolts, these channels, they just contribute to this -70 millivolts, about -5 millivolts. Why? It's because they are pumping 2 cations inside and they are taking 3 cations on the outside. That shows that more of these cations are being taken inside, in as much as there are also some that are being brought in. More of these cations are being taken outside, in as much as there are less that are being taken in. So they are just going to be involved in the pumping of 3 sodium outside and 2 potassium inside, and this will cause the net loss of the cations that will be living inside of the cell. So meaning the cell will be left with a certain level of negativity, which we are saying is -5 millivolts. And that -5 millivolts is what is going to contribute to the rest membrane potential of a neuron, which is about -70 millivolts. So this is one factor. Then the other factor that contributes to the rest membrane potential is the leaky channels. We have what we call the potassium leaky channels. So from the start, we said that these leaky channels are always open, allowing the passage of these ions either going to the outside or going to the inside. But the major factor that determines the direction of these ions, it is the concentration gradient. So you are going to find that these potassium leaky channels, they will be moving from inside going onto the outside. These potassium leaky channels will be supporting the movement of potassium from inside going into the outside. Why? This is because we have more potassium on the inside of the cell compared to the potassium that is on the outside of the cell. So according to the concentration gradient, it will be favoring the movement of potassium from the inside of the cell going onto the outside of the cell. Then what happens here is that when potassium is on the inside of the cell, it is bound to anions. Anions, these are negatively charged molecules or ions. So potassium, when it is inside, it is bound to negatively charged anions, which can be a protein or it can be inorganic phosphates. So you are going to find that when potassium is being pumped onto the outside, it is leaving this negatively charged anion. This negatively charged anion, it is left there. it is going to contribute more to the negativity of the rest membrane potential. So you're going to find that as more of potassium goes on the outside following its concentration gradient, it is going to leave more negatively charged anions inside of the cell. And due to that, potassium will be contributing approximately about negative 88 millivolts to the rest membrane potential. But again, you may ask yourself, how come the rest membrane potential when F cell is about 70, but how is it contributing to negative 88? You are going to see as we finish the whole factors. So what I want you to get here is that the cell is more permeable to potassium than sodium. That is why it has got more potassium leaky channels compared to sodium leaky channels. And due to that, you're going to find that these leaky channels, they support the movement of potassium from the inside going to the outside, because potassium is highly concentrated on the inside of the cell compared to the outside of the cell. And due to that movement, potassium tends to leave more anions inside of the cell, because that is where it is bound. So as it's leaving, it is leaving that negatively charged anion, which is the anion. So as it moves like that, it is going to contribute about negative 88 millivolts to the electronegativity of the rest membrane potential. Then the other factor that I can add on, it is the same anions, the presence of anion, the negatively charged molecules. So you're going to find that these anions, they will be contributing, there will be a factor that will be contributing to the electronegativity, as I have mentioned them. So when you are explaining this in a question, you can either separate them or you can just explain as one, depending on the marks for that particular question. Then the other factor, it is the sodium leaky channels. So these sodium leaky channels, when you just see leaky channels, they will just be supporting the movement of that particular ion. So we know that sodium is more on the outside. According to the concentration, it is more on the outside. So these sodium leaky channels, they will be allowing the influx of sodium into the cell. But we know that when sodium gets into the cell is not going to remain negative, or it is not going to be negative as potassium is being negative. So meaning that the sodium has got a positive contribution to the rest membrane potential. So you're going to find that the sodium will be contributing about positive 23 millivolts. Yeah, about positive 23 millivolts. Then you're going to find that when you add this negative 88, negative 5, when you add them, then you also add this 23, you're going to find that it will take you back to the rest membrane potential of this particular nerve cell. Yeah, so just a minute. Yeah, so these are the factors that help or that contributes to the resting membrane potential of a cell. So when a cell is at rest, that negative 70 millivolts, this is how it is maintained. These are the factors that are involved in maintaining that negative 70 millivolts, so that the cell can just remain at rest, not excited and not again hyperpolarized, or it is not repolarized like that, just like it is just at its resting state. So we have got sodium leaky channels, potassium leaky channels. We have also anions that contributes, and we also have sodium potassium ATPase pumps. So having known this, there is what we call an equation. So this equation, it is known as the Nernst equation. So this equation basically just helps us to understand, or it just helps us to know how much electric potential that does a particular ion contributes, or how much of electrical potential can that particular ion alone would create or would contribute to the membrane when the membrane was just permeable to that particular ion. So what I'm saying is this. First of all, the next equation looks like this. We are going to have the E, which is going to be symbolized in the same electromotive, or we are just going to say the electric potential. E equal to 61, then times the log of the concentration of that particular ion on the inside of the cell divided by the concentration of that particular ion on the outside of the cell. Then you do this. Then in some other books, you will find it being written as E being equivalent to RT over Z, then the ln of the concentration of that particular ion outside over the concentration of that particular ion inside. Yeah. So, as you can see, there is a different setup in how these formulas are. But when you use them nicely, they will give you the same answer. So for this one, when you look at it, we have got this Z. This Z, basically, it is just a number that is showing the valence of that particular ion that you are dealing with. Let's say if you are dealing with sodium, it is just a positive here, and also potassium is positive, so meaning that the valence for these, it is just going to be 1. But if let's say we are dealing with an ion that is having a valence of 2 here, it means that Z will be equal to 2. But in the case of potassium, it means that Z is equal to 1. Then this value that you are seeing there, it is just R, it is the ideal gas constant, and temperature, it is the T, which you are seeing, of which temperature usually we work with it in Kelvin. Yeah. Then we also have this. So if you have seen here, we're having the concentration on the outside over the concentration of the inside, because there is in here, which we can say it is an inverse of log. Then log here, we're having the concentration on the inside on top over the concentration on the outside. So this 61, it is basically a constant number for this particular equation. So most as we are going to be calculating, we'll be using this equation. Yeah, just to bring peace, we'll be using this equation, of course. So as we said, this next equation, it is known as the next equation. It is basically used to determine how much electric potential can one ion, let's say how much electric potential can potassium alone contribute if the membrane was only permeable to that particular potassium, or to potassium alone. How much is it going to contribute? So we're going to find that if we know that potassium on the inside, its concentration is about it's about 40 milli equivalents. Then on the outside, it is about 4 milli equivalent. Then you say calculate how much this guy is contributing to that. So you're just going to basically get this, you put it there, get that, you also put it there. Then you calculate, then you find how much is it contributing to that. So this is just how the next equation works. It just tells us how much electrical potential that ion is contributing if the membrane was only permeable to that particular ion. That is what it is saying. Then the other thing that we need to understand here is the graded potential. So graded potential, these are just small local changes in the membrane potential. The small changes that are going to be appearing in the membrane potential. So we are going to find that the membrane potential will be having small changes. So these small changes, they can differ in terms of the strength of the stimuli. So if the stimuli, it is not that much strong, you are going to find that it is not going to be eligible enough. That potential or that local change is not going to be eligible enough to make that membrane potential to reach threshold and fire an action potential. So what happens in the case of graded potentials is that these graded potentials, they can be depolarizing or hyperpolarizing, meaning that they can either depolarize the membrane potential, or rather the resting membrane of a cell, or they can hyperpolarize it. They can either be ways. So we have got two types of these rather graded potentials. We have got what we call excitatory postsynaptic potential, just from the term excitatory. So meaning that these potentials, they are meant to excite. To excite, it means that they are meant to cause depolarization of the cell so that it can initiate or it can fire an action potential. So what happens is that, let's say we are having a stimuli, and that stimuli can be a neurotransmitter, and that neurotransmitter, it is specifically a excitatory neurotransmitter. For example, we have a neurotransmitter that is known as glutamate. So glutamate is excitatory, is an excitatory neurotransmitter. So when that excitatory neurotransmitter comes in and it binds to its receptor on this channel that we are seeing here, what will happen is that this channel, it will allow the influx of cations such as sodium. When it allows the influx of sodium into this cell, what will happen to the cell? The cell's resting membrane potential is going to start increasing. It is going to start moving towards 0. So for example, if this is the mark for a 0 point, then a molecule, which is an excitatory neurotransmitter, goes and binds to this channel, and this channel, they start allowing the influx of sodium on the membrane of a cell. You're going to find that if the resting membrane potential of a cell was here, this is going to start moving because the negativity of that cell has started being overcome due to the influx of what? Positive cations, because we saw here that as sodium was getting in, it was contributing about positive 23, but in as much as it was contributing, that it was not significant enough to cause the resting membrane potential to be positive, but it remained negative. So in a condition where we are having the opening of these channels that allow rapid influx of sodium due to the binding of an excitatory neurotransmitter, you're going to find that the inside of the cell will start becoming positive. So as it starts becoming more positive, it will start moving away from this resting membrane potential towards 0. So you're going to find that if this excitatory neurotransmitter, which is glutamate, it is not that strong enough, or the stimuli is not that strong enough, these the depolarization won't reach it won't reach the threshold potential. So meaning that it will fail, it will go down. Again, it might be initiated. Again, a stimuli might come, and then it will initiate it. Again, if it is not strong enough, it will go down. That's what happens. But you know you want to fire this action, but you want an action potential to be fired. So what happens is that there is going to be a mechanism that will come in that will cause these excitatory neurotransmitters to initiate an action potential in this scenario that we are talking about. So before we get to go there, let me also explain about the other type. Then the other type of graded potential is what we call inhibitory postsynaptic potential. See, so this, what they do is that when an inhibitory neurotransmitter comes and binds there, for example, we have inhibitory neurotransmitter like GABA. When it goes and binds to this receptor, this channel will open. What this channel will do is that it will either allow the influx of anions, like negatively charged ions like chloride, or it will allow the efflux, the movement of positively charged ions like potassium moving out of the cell. So the purpose of this inhibitory is to inhibit this cell not to fire an action potential. So meaning that if the resting membrane potential of that particular cell was here, when there is a binding of an inhibitory neurotransmitter, this resting membrane potential even become more negative, because we are going to see more influx of negatively charged ion, or we are going to lose more positively charged ion. So meaning that the inside of the cell is going to become negative, because we have understood that this factor contributes to the electronegativity of the resting membrane potential. So now, in a condition where you wanted to fire an action potential, but you cannot fire it because the stimuli is not strong enough to cause the resting membrane potential to reach threshold, there's a mechanism that comes in, or there is a concept that works there that we call summation. So summation is basically just the addition of these graded potentials to determine whether the neuron will reach the threshold potential. So this summation, what we'll do is that these excitatory postsynaptic potentials are going to be fired. When they are fired, they are going to be summated, and when they are summated, when they are added together, the stimuli is going to be strong enough to fire, or to cause the membrane to reach the threshold they need to fire an action potential to depolarize like that. Then the other part is for the inhibitory, like that. It will also undergo the same summation, but we mostly focus on the excitatory part. So what will happen here is that there will be summation. So now summation can be in different types. We have got the first one that is known as spatial summation and temporal summation. So what we have to know about this is that spatial summation it involves multiple neurons stimulating one neuron at once or simultaneously. They are going to be firing their stimulatory stimuli at once, and the effect of these stimulatory stimuli is going to be added together and fire an action potential. So you are going to find that you are having one neuron here, and this neuron, it is connected to multiple neuron. It is connected to other multiple neurons like this. So under spatial, you are going to find that this neuron will fire, and also this neuron will fire. They will fire simultaneously. They will fire at once, and their effect on this neuron, it is going to be summated there in the cell body, and there will be initiation of an action potential at the axon heel lock. So I'm going to find that when this one fires, it will move maybe up to this point. Then they will add on the effect of this one. Then the other one, it has even reached threshold potential, then they'll be firing. So spatial involves multiple neurons firing at the same time or spontaneously, or rather not spontaneously, but simultaneously. They are firing at the same time at once, and their effect will be added together, and that will cause the rest membrane potential to depolarize, which is bringing about the action potential. Then for temporal, what happens is that one neuron will be stimulating the other repeatedly. So meaning that one neuron, if this neuron is only connected to one neuron like so, it means that this neuron here will be stimulating this simultaneously. It will be stimulating the other one repeatedly in succession. Not simultaneously, but it will be stimulating the other one repeatedly in succession. Like it will fire an action, a stimulatory potential, another one to fire stimulatory, another stimulatory to fire. So these that it will be firing, they are going to be added, and as they added, the effect will build up. That will cause the rest membrane potential to reach threshold. Then from there, it will fire an action potential. So undergraded potential, we have got excitatory postsynaptic potentials, which are excitatory. When they are released, they will cause the depolarization of a cell, of the cell membrane. Then we also have inhibitory, of which when they are released, they will cause inhibition, which is bringing about hyperpolarization. Then under these, how they work is that they undergo summation. So it can be spatial or temporal. Spatial, they are coming from multiple neurons at once, and their effect is being added to initiate the action potential. And temporal, it is just from one, meaning that one will start firing these stimulatory stimuli to stimulate one neuron, but this stimulatory stimuli will be fired repeatedly, and their effect are going to be added together. Then it will build up, and they will be firing of an action potential depolarization. Yeah, so basically, this is the whole concept that is behind that. Now, right now, you may ask yourself, okay, since now we have learned all that, how is it now applying? That is because, as I mentioned earlier, a cell or a nerve cell, basically that we are talking about, it is the one that is found in the nerves that innervate these muscles that we have, and those cells that are found in the muscle, they have got the rest membrane potential that we are talking about. So these nerve cells, they are just going to be involved in the transmission of the signal, taking them to the effector organs, like the way we said. So the nerve cell here, as you have seen, it has got these different parts. You have got the soma, which is the cell body, the nucleus, the dendrites, the myelin sheath, the axon, the nodes of Ranvier, as well as the axon terminal. So these parts, they are going to have their roles that they will play in the transmission of this action potential. So more of how now these neurons, they will transmit that signal and bring about the contraction of that particular muscle or the movement of the limb due to the contraction of the muscle. We are going to understand it further. So right now, the concept that I just wanted us to build is the same concept for the depolarization. What happens if the cell is depolarizing? It means that the inside of it is becoming more positive and less negative, meaning that they'll be firing of an action potential due to the depolarization, and so on and so forth. Then the other thing that I just want us to have a brief understanding about is what we call the curve, the depolarization and repolarization curve. So just to have a quick review on it. So as we said, we have got the resting membrane potential, which is at this part. Then let's assume this is our threshold potential. And then this is going to be our 0. Then this is going to be, let's say, our positive 35 millivolts, where it's going to be the highest part. And here, the resting membrane potential is negative 70 millivolts. And maybe the threshold, it is negative 50 millivolts. So what happens is that when the stimuli is sent to that particular cell, the resting membrane and it reaches to a cell, it gets to bind to those channels and allow the influx of those channels. At first, what will happen is that this cell, the resting membrane potential of a cell, will cause the opening of those channels. They'll start allowing small influx of cations or positively charged ions. When they reach to the threshold point, there's going to be rapid influx of positively charged ion, which is sodium. There will be rapid influx of sodium. So rapid influx of sodium will cause rapid depolarization of that cell. So meaning that the depolarization of the cell will shoot. It will shoot, and as it reaches its peak, those channels that open, they are going to close. So at this point, what you have to understand is that when the resting membrane potential of the cell gets to reach at the threshold potential, which is negative 50 millivolts, what happens is that the first sodium channels, they open. So these sodium channels, they have what we call activation and inactivation gates. So what happens is that at rest, or when the cell is at rest, it is at its normal resting membrane potential. These activation gates, they are closed, and the inactivation gates are what is open. The activation gates, they are closed, which are going to be these gates on top, for example. Then these inactivation gates are going to be open. So these inactivation gates are the one that allows the small sodium to be entering so that they can help in the maintenance of the resting membrane potential. So now when the resting membrane potential depolarizes, it reaches at this point, it reaches at the threshold point. What happens is that these sodium voltage gated channels are going to open. When they open, what opens is these activation gates. So these activation gates of this particular sodium channel, they are going to open. And as they open, they will be allowing now rapid influx of sodium into the cell. So the sodium will start moving in rapidly. It will start moving in rapidly. The sodium will start moving in rapidly. So as it will be moving in rapidly, to cause the inside of the cell to become less negative, approaching the positive side, because we're having too much of sodium that will be entering. This is why we are seeing the curve rapidly shooting. And at this point, this is the point that we are calling depolarization. So when they reach at the peak at this point, because we know that sodium, when it is just entering, its target is to reach what the membrane potential of about positive 60 millivolts. But when it reaches somewhere about 35 to somewhere 45 or 40, what happens is that these inactivation gates, or rather these activation gates, they start closing slowly. So meaning that the activation gates that were opened, they will start closing slowly. So when they start closing slowly, they will be decreasing the influx of sodium. Together with these inactivation gates, they will also be closed when it reaches at this point. So meaning when this channel is closed and this channel also closes, there will be no more influx of sodium. And what happens is that on the other hand, the potassium channels, they will open. The potassium voltage gated channels, they will open, because they will sense to say the resting membrane potential has reached its peak. So the potassium channel will open, potassium voltage gated channel. Due to the change in the voltage, they will sense and they will open. When they open, they will allow the influx of potassium. Potassium will start moving from the inside going onto the outside. And we know that when you are losing these positively charged ions going onto the outside, the inside of the cell will start becoming negative, or more negative and less positive. So that means that the cell will start now approaching towards its normal resting membrane potential. So as it is going back, its resting membrane potential is now slowly being restored. And we said that the restoration of the resting membrane potential is what we call repolarization, which is just due to the efflux of potassium. And here it is just due to rapid influx of sodium, like that. And now as it starts going, we know that these potassium voted gated channels, they are quite slow when it comes to closing. When they open, they take a little bit of time to close. So instead of allowing the required amount of potassium to leave, they will allow even a little bit more or extra potassium to leave. And as more potassium leaves than the normal that is supposed to leave, it will cause the resting membrane potential to hyperpolarize, to become more negative. So as it becomes more negative, the sodium ATPase channels will come in. They will say, no, this is not how the resting membrane potential will be. So those channels will start now working, pumping 3 sodium out, 2 potassium in, so that they can maintain the normal resting membrane potential. And these, as that happens, the normal resting membrane potential will be restored, and the membrane potential start moving back to normal. Then it will come back to normal. This is how it moves. So this is basically what happens. So meaning at the point where there is firing of an action potential depolarization, you're going to find that your muscle, that's the point where it will contract. When there is repolarization, that's the point where your muscle start now relaxing. You get the point, right? So more of that data will be built on, but I just wanted you to understand the basis of how the resting membrane potential, the action potential, the graded potential are going to help us to understand how these they apply in the contraction, relaxation of the muscles, as we are going to go deep, deep in. So this is basically the whole information. Otherwise, if there are any questions, you are free to ask. You can post in the group, you can inbox either of us who are helping you into the whole thing, and please comment so that we can know where we can add on and where we can focus on the most. Otherwise, thank you very much for your time. Have a blessed evening.